So we'll continue with our second talk. Our second talk is from Kuhl, Stanford University, Data-Driven Modeling of COVID-19, Lessons Learned.
Yeah.
Ready? I'll turn it over to you.
Thanks, James. So yeah, I'm scales way up from what you just presented. So I'm going to talk about population modeling of COVID-19. So I'm going to try to explain what we did to approach this and what we learned actually from doing this throughout the last year. So you're probably all familiar with the compartment models that people use to understand the spreading of epidemic diseases or outbreaks. And we use here the SCIR model, which is the model with 4 compartments, just to repeat that for everybody. So we have a susceptible, exposed, infected, and recovered group. And the population transitions between these 4 groups with 3 rate constants. So that's the contact period. That's how people come into contact with one another. The latent period. So that's for people that don't yet show symptoms and are infectious. And then the infectious period is when people can infect one another. And the ratio between the contact period and the latent period is what we all know as the reproduction number. I think by now everybody knows this because it's been in the news quite some. So the reproduction number above 1 is an outbreak of the disease, and below 1 is actually that the disease would die out. So we did this pretty early in the pandemic. So we started out in mid-March and looked at different countries. And this is what we did for Germany in June. And this was nice because by then it had gone through this entire first wave. And you can see the infectious group, which is the orange dots from the case data, is already back to pretty much 0. And so from that, we can then fit it to the real to our model data, and you can see it's a pretty nice fit. So the orange dots are fitted to, or the orange line is fitted to the orange dots, and from that we can gain these parameters. So we did that actually in mid-April for all countries in Europe, and I'll tell you just in a second why we did this. So you can see different behavior already here. So there's countries, remarkably, for instance, the last one is Sweden. Sweden never implemented any politics to restrict the spreading of the virus. So it's actually a pretty steep increase in Sweden of a number of cases, whereas, for instance, the first one is Austria, that had a pretty strict policy, and the curve already bends down at the same point in time. So this was neat, and we could then plot that across Europe, and we can plot different parameters. So for instance, here on the right, you see the basic reproduction number. So that's the number that I just introduced. There's also parameters here like the initial community spreading. That's something we don't see. We can just extract from the model and the data. So this is something that happened underneath the first surface before the first case was recorded. And then in the middle, you see how well it was contained. So that's the affected population with respect to the total population of a country. So this is quite interesting for us to study and learn. But then we thought, okay, we actually usually in our pre-COVID life, solve partial differential equations in our lab. And so we thought, okay, maybe we can use that. It turns out that actually it's quite interesting to look at the spreading of COVID, and you can do that with discrete network models. And this is what this is showing. So we create a network where we mesh all the different countries, in this case, Europe. And we do this with a matrix that is actually the graph Laplacian, and the weights of the graph Laplacian are the passenger air travel between different places. And you can see here in this graph in dark red, that's Germany, Spain, France, and Italy. So that's the most traveled countries in the European Union. And then there's others that are traveled less. And the thick lines are the thick connections. So that's the locations where people travel the most. So say from Spain to Germany to France. And that's the edges in our graph. So with this network, we can then solve this SAR model that I just introduced locally for every country, and we can see how populations mix. And this was interesting because when we did this, Europe was in this discussion of whether they should open their travel restrictions. And you probably know that Europe, the entire European Union, actually closed its borders on the 3rd week of March entirely for external travel. So all the colored countries here were cut out pretty much from the rest of the world. And so then this was when they were saying, okay, when should we reopen this? And that's when we did the simulation. So you can see on top a simulation with the travel restrictions in place. So that's what actually happened. And we simulated it with our model. And then underneath you would say what we studied what would happen if there were no travel restrictions. And you can see there's a much larger red, so a larger infectious population especially in Central Europe, Spain, France, Germany, the Netherlands. Whereas if with the travel restrictions in place, you can see everything is more blue and actually less spread. So this was interesting. Was actually, the study was in the news because it was just about when Europe was about to open. So this was got a lot of coverage in Europe. But this was also at a point in time when many models were criticized. And that was because they were actually just not suited for this kind of outbreak. And so what we decided to do is actually introduce something that would fix this error. So you probably remember that there was a lot of discussion about models, a lot of criticism, and all of a sudden, all the models were considered just useless. So this is when we thought, okay, why don't we introduce a dynamic contact behavior that depends on political measures? And that's what you can see here in the middle. We introduce a dynamic contact rate beta, and we make an ansatz that is a hyperbolic tangent function that you can see on the right in this plot. So it starts out with the basic reproduction number R0. It gradually transitions to the reproduction number in lockdown RT. And the time by which it transitions is this T star and T. So T star is when this happens in time, and T is how quickly this happens. And so we have 4 parameters that all have a meaning, and we can actually fit them and see how they relate to politics. To do this now, it's no longer easy to do by hand. And so we consult our friend Tom Bayes and the Bayes theorem that you can see here. So essentially what we do is we make prior assumptions for the parameters. So that's the turquoise color. And we assume a likelihood, so that's orange. And then the output is a posterior distribution of our parameters. This was when we submitted the paper on the effect of this tangent function in Europe. And you can see the same countries here. On top on red, you can see the reproduction number or the contact that starts high. So for instance, for Austria at 4, so that's a reproduction number of 4 at the early outbreak. And then it goes down to something below 1, and that makes this curve actually bend. And so we submitted this paper, and then we said, can we predict it? So all you see in gray here is a prediction, and we predicted 3 scenarios. The first one was leaving it closed as is. The second one, the light orange, is gradually opening, and then the orange, the dark orange, is opening immediately. And then while the paper was under review, actually we run the time went on, and we had these dots that we could then in retrospect retrace. So what you see on the gray on every graph is what happened after the submission of the paper that we had made these predictions. And you can see that most countries actually follow the behavior in complete lockdown. So most dots in the gray window actually lie on the prediction by just continuing lockdown. So then we thought, okay, what does this reflect? And we have this nice function, what can we do with it? So you probably know that air traffic dropped substantially, in fact, in Europe by 95% within only 10 days in the middle of March. And when you look at the air travel, and that's the dots that you can see on these plots, it takes the same kind of functional form as our reproduction number. So it has this tangent type function where it starts at 100%, goes down to 5%, and there's this transition. So they look actually quite similar. So again, the dots are the air traffic, the red line is the reproduction number, and the lag between the two we call the responsiveness. And that's something we learned from the data. We did this not only for air traffic, but also for cell phone data. So you probably remember there was this big publication or publicity of Apple. They recorded the mobility data of people during COVID. So we used those data, and that contains driving mobility, walking mobility, and transit mobility. And then we had the air traffic. And all of them follow this kind of tangent curve that you can see there. And on red is the fit of our reproduction number, and there's the responsiveness. If you do this for all countries in the European Union, you can see that the mean responsiveness is 17 days. So that's a little over 2 weeks. And you can also see that different countries responded differently. So if you look at the last column in this graph, you can see Sweden on the bottom, and I already mentioned Sweden. So they didn't really do a whole lot, and you can see their responsiveness is much, much slower. So it's about 24 days. And then there's other countries that responded on exactly 2 weeks and some much faster. So this is kind of interesting because it actually does relate to the politics in different countries. This was at the time when the first people talked about asymptomatic transmission, and it was actually back then pretty critical, and people didn't believe in it. And it was interesting because we teamed up with a group of people that are on this paper that's listed below. So that's Jay Bhattacharya, Aaron Ben David, and John Jordanis. And they're all at Stanford, and they did the first seroprevalence studies in Santa Clara County. And you can see that listed here as one of the 9 locations that we analyzed. And that was by mid-July. And what the study found is that there was an undercount on the order of 20 and above. So what the undercount means is that we count 1 person, and in reality, there's 20, where 19 are asymptomatic. And so that's quite a huge number. So we thought, okay, we can't really confirm this. This was all over the news because people were very critical about this number. And so we said, okay, can we at least visualize this? We can't see it, but can we visualize it computationally? And this is what we did. We added another box to our model. So this is this turquoise box there. And so what this does is from the exposed group, from the red, people can either become symptomatic or they become asymptomatic. And that's our only distinguishing parameter. So that's a fraction of symptomatic to asymptomatic. And that's essentially the undercount. And from that, both recover. So these 2 disease parts can have different disease dynamics. So it can be infectious for different times, for instance, because they're actually simply more outgoing, more infectious, they cough more. So there might be different dynamics individually, but we can actually derive all the equations analytically for these for this extra 5th group. We can also just fit it to the data. So this is for the Netherlands, and if you remember, it has an undercount of 5. So for every healthy person, they counted 5. For every symptomatic person, they counted 5 asymptomatic. You can fit the orange curve, and we can predict what the asymptomatic population would look like and how the total recovery would look like. So in this case, of course, much more people would have recovered than we would know from the databases that we look at. So that's interesting, and you can do this for all places. So for instance, Santa Clara County, so there was the first study. New York did a study where they just studied people that walked into grocery stores, took their blood and did seroprevalence of them. So that was one of the first. And then there was also a famous one in Germany in Heinsberg. And so we compared all of them and we found they all look pretty similar. So all have kind of same dynamics. So the first one here is the reproduction number, the red plot. And then underneath you can see these different boxes color coded by their box. And you can see that the asymptomatic and recovered group look pretty similar for all these groups. We can also actually, because we do Bayesian statistics, we can go back and say, hey, when did the first person show up with the disease that was not properly recorded? So in Santa Clara, we found that would be on January 20th. And we found that actually way before it was confirmed that there were cases before the case that was reported on February 23rd, I think. So this actually matched that quite well by doing Bayesian statistics. So then we were contacted by someone, and this was actually the general attorney for Newfoundland. And Newfoundland is a province in Canada. In fact, it's the smallest province. And you can see it here as the center of this network. Newfoundland is Labrador and Newfoundland, and Newfoundland itself is an island. And so the island had at some point in May, no cases of COVID. And so they had closed everything. So they had completely cut all the connections you see here to air travel. And so then their general attorney was actually taken to court because someone challenged the travel restriction. And so then they came to us and said, can you simulate what would happen if we reopen? So we did that. We had 3 different network models that you can see here. Red is for all of North America flying to Newfoundland. And then light blue is for only Canada. And for dark blue, it's only the Atlantic provinces close to Newfoundland. So that's only 4 provinces. Again, we create the graph Laplacian from air travel statistics and then simulate the outbreak in all of North America and the people that come into Newfoundland on a day. And this is the plot. So the color code here tells you how many people from that state would come to Newfoundland on a given day and be either exposed, so in the red bin, or infectious on the orange bin. So these are the 2 different plots. We can just add that up and we can count how many people would come to Newfoundland and bring the disease. So this is less than 1 per day. But if you open, on average, there would be some people within a couple of weeks and they would then induce the disease. So this is the same thing in just a plot and with statistics. So on the bottom, you see the different network models. So the North America, Canada, and the Atlantic provinces. And on the top, you see what would happen if people would perfectly quarantine by 50%, and quarantine by 0%. And so we can quantify what would happen at which day if they would reopen the country. And what happened then is, this is Proton Rahman, one of our co-authors. He testified in court and we actually won the court case. So there was actually decided that it was the right decision to keep Newfoundland closed. So this was the very last real paper. And then by the end of the summer, when this court case was announced, we offered this class actually at Stanford. And the idea of this was that we were thinking, why can't our students calculate what would happen if we reopen campus? So you're probably all aware that there was big discussion in the fall, a lot of campuses keeping closed. In fact, Stanford is not yet open and has been closed for a full year to undergraduates. Many residential campuses have been very controversial in opening or had just closed right after again. So we said the students can calculate this themselves. We make a class and we give them a grade and they have to come up with a solution.
I'm sorry, 5 minutes.
So here's a group of our students. And so we challenged them to come up with a model. And they first looked at the data. So this is some of the results. You might recognize your institution here. If you don't, it's probably because they didn't have a high number of cases. If you do, and it has a high number of cases, don't worry, it actually most of them managed the disease quite well, which was not clear at the beginning of the fall quarter. So you can see Notre Dame has a high number, Wisconsin, UIUC, University of Florida, Clemson. So these are all campuses that have 4,000 cases or more. And this is the incidence. So the incidence, by now, I guess everybody knows what it means. So we plotted as a 7-day incidence. So we take the 7-day sum of cases and divide it by 100,000 people. And you can see that smaller campuses now, like Notre Dame, actually pop up because they have a higher incidence per student body. And then Wisconsin, Clemson, and Arizona had the peak cases. Here's a plot of the campuses that we looked at, a total of 30. And you can see in green, the case numbers increasing and then decreasing again. In red on top, you see the reproduction number, and in the bottom, you see a table. And I would like to direct you to the last 3 columns. So the third to last column is the recovered fraction, and that means how many students actually had the disease throughout the fall quarter. So say Clemson has 0.22, which means 1 out of 5 students had become infected throughout the fall quarter. And then the last 2 columns are the days to campus peak from campus reopening and the days to county peak from campus peak. And this is interesting because you can see them in this graph for 30 campuses. So the dashed line always represents the opening of the campus. The red number is the number of days until the campus saw a peak. And all of these are within 2 weeks. And then the blue number means that the campus peak leaked into the county. So actually that's kind of our definition of super spreading. It's not just creating a lot of cases, but it's also spreading into the county. And the red number also sees actually peaks within a week or 2 from campus peak. So this, as you can guess, got a lot of coverage because people were just interested in seeing whether campuses are super spreaders. So it was all over the news. And this was in a way just an analytical study to show, okay, within 2 weeks, the campus sees high numbers, can bring the numbers down. It leaks into the county, but the county for most places couldn't bring the numbers down as drastically as the campus itself. So it was kind of good and bad news. So with that, I just leave this up here. I don't think I need to summarize this. I'm happy to discuss things in the meeting. I think our main takeaway from what we've done is that we can model the dynamics with dynamic parameters, and we can model the control and correlate the control using actually what we've just shown and correlate that actually to political actions. And that can help, obviously, to select successful reopening strategies. And I'm happy to answer questions in the after session. These are the people who contributed it. Thank you.
Okay, thank you very much. Really appreciate that. We have time for 1 or 2 general questions to either of our speakers. People feel free to speak up. If anyone have a hand raised, I'm looking, hold on. Jacob, please.
Okay, this is actually a very interesting one. You actually noticed that the numbers reported are incorrect. This is something which is interesting because this is something that needs attention, and you did pay attention to this. You said that each reported case is worth 5. This is where, is this the number you used? Did you try multiple of those values?
You mean the asymptomatic transmission?
You said that there was a publication that says that each reported cases were 5, and then you incorporate that number, if I recall your talk correctly.
That's right. So for the Netherlands, so this is based on blood samples, right? So this is seroprevalence. So people did blood sample tests, how many people had already had the disease. And so the one that I showed was the Netherlands, where this is about 5 to 7. There were also other studies that reported much higher values. For instance, if you look at Brazil had 5 to 27, or Idaho 10 to 17 times. Those are numbers, of course, during the early pandemic. So I would think now these numbers go down because we test a lot more. So we identify asymptomatic cases. So for instance, for the campuses, most campuses test every student every week, as you know. And so there, these numbers I would expect they would be like on the order of 1 or maybe 1.5. Yeah, good question, actually.
There is a continuation, but we'll talk after.
Great, yeah.
Other questions? I had one question myself, because we, of course, like everyone else in the mathematical biology class, did this kind of modeling, not with the kind of effect that you did, which was that you show the active case numbers. But if you look at how the active cases are determined, they look at it at time of incidence, and then very often they simply assume that the case is cleared after 3 weeks. And so that the active case reported number is usually not a reliable number. Were you actually fitting active cases, or were you only fitting new cases for hospitalizations?
That's actually a very good point. Let me just share this again, and you can see exactly, for those of you not familiar with the problem. So problem is really that we try to fit the reported cases and the recovered cases. And if you try to fit the recovered cases as well, so that's the parameter C here, then actually you end up getting very long periods. And that's really a problem in recording. And that we try to first initially fit the data to the reported case. And we realize, man, this is like 3 weeks. So nobody is infectious really for 3 weeks. And we know this from the disease and from the way the disease is reported. This is really a reporting issue. And so then we went back and really from then on only used, here you can see that the blue dots are not really fit by these curves at all, right? And that's exactly the recovered. But then we started to just actually only use the infectious population, and that worked quite well, and use clinical values for the length of the disease. Good point, yeah.
Okay. Anyone else? Anyone from our silent audience have a question or two?
I do have one question if there is time.
Please.
Thanks. Thanks to speakers. This is Asan Krazmian from Brown University. So I have a question for Ellen about the model that you're using. So I have seen so many people, so many different papers that they are using different models. So the question was how you trust the model that you're using, especially that you said that, for example, you started with the SEIR model and then you got a lot of criticism and then decided to add the asymptomatic into it. And I've seen so many different variations of the model. So, so which one is the best to use? So do you have, in the lessons that you have learned, can you share with us which model to pick? Thanks.
That's actually a good question. I see a lot of people nodding. So I think that's the golden question, actually. So what we've done is we have naively started, so I'm not an epidemiologist. So we've started this, like James said, with just very simple models, like we do in mass bio. And then we have gradually added things that didn't work. And I think that's in a way how modeling in general should be. I mean, you start simple and then you add things and add complexity. And then as you do that, you actually learn more about how the models work. And I wouldn't say there is the single one model that could do everything. There's another approach, of course, right? There's people who come with like a gazillion types of parameters, and they have huge models. You can model every population, every age group, right? People have different age groups. How do they interact? And everybody's like, oh, wouldn't it be nice if you could do this? Yeah, sure. It's easy to do, right? I mean, you can add more equations, you can add more parameters, but the problem is, how do you define them? And I think James alluded or asked that question initially, right? I think what we have now is a model where we know what the parameters do and how we can define them confidently from the data, but we can probably not model things that are really parameter driven, although it would be possible technically to do. But I think that's just the art of modeling. And I'm not claiming we know how to do it, but I think that's the golden question that everybody has to address.
All right, thank you very much.
Okay, I hope some of our audience will be able to stay on and have more intensive conversations with our speakers. Two very different and both very stimulating talks.